Today we're going to talk about measuring hormone levels, what controls the levels of hormones in the body, and how we can actually measure what those levels are. The blood level of a hormone equals the amount that's produced by the endocrine glands, the specific sites of production, minus the quantity of the amount of hormone that's degraded and the amount of hormone that's excreted. So what sorts of things degrade hormones? How do hormones get degraded? One of the primary sources is digestion in the gut. As hormones get into the gut, they're digested just like other organic compounds are in the gut and lose their activity or are changed in form. There are enzymes within the blood that degrade some hormones, so actually when they're secreted into the blood, they actually get degraded over time by the enzymes that are actually in the blood itself. The primary source of degradation is liver metabolism. One of the major functions of the liver is to break down compounds, to detoxify compounds in the body, to put them in a form that can be excreted. The liver contains a wide range of enzymes that are capable of breaking down many different kinds of organic compounds. So the liver metabolism can alter the form of the hormone, change it from one form of hormone to another form of the hormone, or they can actually take it apart, just destroy it, turn it into carbon dioxide and water. How are hormones secreted? Well, essentially every way that something gets out of the body, hormones go out that way. So we find hormones in the saliva, in the feces, in the urine, in the sweat, and even in the tears. So any way that something leaves the body, it will carry hormones with it. Protein and peptide hormones are found in the saliva, although it's very difficult to measure them. They're found in very small amounts. They're excreted intact as the hormone itself in the urine and in the feces. Steroid hormones are present in all excretory products, so they're found in the saliva, feces, urine, sweat, and tears. Steroids have a specific mechanism by which they are altered. This is called conjugation, which we'll talk about more in a moment, which improves excretion of these hormones from the body. Excretion of steroid hormones are a special problem because steroids are lipophilic. Lipophilic means fat-loving, and they're hydrophobic, which means water-fearing, or it means they are not easily dissolved in water. They're easily dissolved in fat or oil. Steroids are therefore not soluble in water, and cell walls are essentially lipids, or fats, so steroids easily pass in and out of cells. There's no active transport mechanism in and out of cells for steroids. They just passively diffuse into cells and out of cells. The excretion pathways, steroids in their active form pass through cell walls, which means that they have access to the blood, they have access to the urine, they have access to feces, and that's why we can find steroids in saliva, sweat, feces. Small amounts in their active form in the urine because they're not water soluble. Large amounts don't actually get into the urine, but some small amounts do get into the urine in their active form. And then, as I mentioned before, they're actually found in tears in their active form. However, to improve the excretion of steroids, steroids are conjugated, and conjugation makes them hydrophilic or water-loving. So this makes steroids water-soluble so that they're easily excreted. Let's talk a bit more about steroid conjugation, what the process is. So only steroids that have a hydroxyl group can be conjugated. So any steroid that has two ketones, for example, cannot be conjugated because it requires a hydroxyl group in order to be able to become conjugated. Conjugation requires an enzyme to attach the conjugate, and these enzymes are found in the liver, the gonads, the placenta, and the adrenal, and they're specifically for making these steroids water soluble through conjugation. The conjugate turns the steroid from a lipophilic hormone to a hydrophilic hormone. It now is water soluble, which means it can easily get into the urine and it can be excreted. And the vast majority of conjugated hormones actually are excreted through the urine. The effect of conjugation, not only does it make it water soluble, but it actually inactivates the steroids. So conjugated steroids are not biologically active in the way the unconjugated steroids are. It simply is a mechanism designed to make it possible to excrete steroids from the body easily. There are, however, other enzymes in the body that can remove the conjugate, which will make the steroid active again. So there's this kind of interplay where some enzymes facilitate the conjugation of steroids. Other enzymes can take conjugated steroids as their input. They can remove the conjugate and release the active form of the steroid back into the body. So unless they've been excreted, conjugated steroids essentially can be turned back into active steroids at another time. There are basically two families of conjugated steroids, two types of conjugated steroids. There are the glucuronidase. These conjugates use glucuronic acid, which is a derivative of glucose actually, and it's attached where the hydroxyl group is on the steroid molecule. So here's an example. Here's testosterone. I've circled the hydroxyl group on testosterone. That's where the conjugate's going to be attached. And this is testosterone glucuronate or testosterone glucuronide. Both names are appropriate. You'll find them referred to in different ways. But you can see this residue of glucuronic acid, molecule that's smaller than the steroid, but not inconsequential molecule, is attached where that hydroxyl group was before. The second family are the sulfates, where a sulfate group is attached where the hydroxyl group is on the steroid molecule. This is probably the most common sort of conjugate. Large amounts of steroids are conjugated as sulfates. Here's an example. Here's estrone. I've circled the hydroxyl group in estrone. And this is estrone sulfate. You can see where that hydroxyl group was. There now is where the hydroxyl group was, there now is a sulfate attached to that. This is now no longer active as an estrogen, this estrone sulfate, but it is highly water soluble, and large amounts of this are excreted. Dehydroepiandrosterone, one of the weakest androgens, but produced in high quantities, is very rapidly sulfated in the body and is excreted in the urine as DHEA sulfate, and probably the largest amount of hormones that are excreted are in the form of DHEA sulfate. How do hormones circulate in the blood? Protein and peptide hormones circulate in their active form and are not bound to any other compound. In other words, they circulate in an unbound or free form. Steroids, on the other hand, circulate in three different forms in the blood. They circulate either as conjugated steroids, so for example, DHE sulfate, estrone sulfate, testosterone glucuronide. Steroids also circulate in their unconjugated form, bound to steroid binding proteins. In fact, about 95% of steroids circulating in the body that are not conjugated circulate in a bound form where they're attached to a protein. And we'll talk a little bit more about that in a moment. Only about 5% of steroids, unconjugated steroids, circulate in their free form with just the steroid molecule by itself. Here are some examples of binding proteins, which we'll talk more about in later sections of the course. Alpha-fetoprotein, primarily present in rats. This is a protein that binds estradiol. but doesn't bind testosterone, so it's very exclusive to a specific steroid. SHBG, or sex hormone binding globulin, is found in primates, including humans, and now there's evidence that it's actually found in rodents also. This compound binds both testosterone and estradiol, as well as dihydrotestosterone, but it binds the androgens testosterone or dihydrotestosterone better than it does estradiol. And we'll spend quite a bit of time later talking about the possible role that this plays in adjusting how much free hormone there is circulating in the blood. Corticosteroid binding globulin, or CBG, binds corticosteroids, very large amounts. Most corticosteroids circulate in their bound form. And albumin is a very common binding protein. It is it essentially binds all steroids, but it binds them very weakly, and there's actually a debate whether a steroid bound to albumin is really not the same as a free hormone because it becomes dissociated from the binding protein so rapidly. One point I want to make about binding proteins is that once a steroid binds to a binding protein, all this binding has a binding constant, which really determines on average how long the steroid stays bound to the binding protein. It doesn't stay bound forever. It may stay bound for minutes or seconds. It depends on how strong the affinity is of the binding protein for the steroid. Another point about binding proteins, which is somewhat unresolved, is whether the steroid, when it is bound to the protein, is completely biologically inactive. And generally, in most cases, it appears that binding to a binding protein inactivates the hormone, keeps it from, it certainly keeps, in the case of steroids, steroids from entering cells. Once they're bound to a binding protein, they are too large to get in cells. They can't pass through the cell walls anymore. So clearly a steroid circulating in a binding protein is not going to be able to enter cells and have biological effects within the cell. So here is a sort of cartoon of how steroid hormones circulated in the blood. The majority of steroid hormones in the blood become conjugated and are put into an inactive form. These are the sulfates and the glucuronides. And then most of those are excreted. The rest of the hormones circulate in an unconjugated form. This is the active form of the hormone. These are the steroids that are capable of entering cells and inducing biological activity. Of those unconjugated, about 95% of them circulate protein bound, so that they cannot get into cells, and about 5% are circulated in the free active form, which easily pass in and out of cells. So the general view here is that the body actually produces large amounts of hormones, many of which are inactivated or temporarily inactivated, and only a small fraction of them actually circulate in the blood in a form that is biologically active. Now, of the conjugated steroids, as I mentioned, there are actually deconjugation enzymes that can remove the side groups off of the conjugated steroids and turn them into unconjugated steroids. So these examples of these enzymes are sulfatase, which removes the sulfates, and glucuronidases, which remove the glucuronides. So the end result is that, for example, estrin sulfate, when sulfatase acts on it, it releases it as estrone, and now it is an active form of the steroid, and it can have biological activity. Bound steroids are considered potentially active. No enzymatic activity is required to release them and allow them to bind to their intracellular receptors, the receptors on the cell of the target tissue. But when they are actually bound to the protein, they are inactive, but they can become unbound from the protein, and then their activity returns. As I just mentioned, conjugated steroids have to be deconjugated to become active again. So how do we sample hormones? How do we get samples that we can actually measure hormones in, and what reflects what's going on in body? In general, um, the blood levels of the hormones, since hormones are essentially defined as compounds that circulate throughout the body in the blood levels really represent the hormonal status of the individual. But there are a number of other sources of samples that we can measure that give us an indication or serve as a good proxy for the blood levels. Even if we're interested in blood levels, there are different ways of collecting samples that allow us to measure blood. These are all invasive, and they have to invade the body in some way. One method is the blood spot. This is what diabetics use to measure blood glucose levels. It's minimally invasive. You use a little stylet here that you simply prick the finger, massage the finger to create a blood spot, and then use filter paper and collect 2 or 3 blood spots, and the hormones go with the blood. You get it takes a small volume of blood, 1 to 2 drops, and you can assay multiple hormones. From that blood spot by taking little punches out of the blood spots and measuring each one of those. The difficulty with blood spots is that because the volume of blood is so low in them, it's very difficult to measure hormones that are in very small amounts in the body. This works for hormones that where there are large amounts, but that works for testosterone, it works for estradiol, it works for progesterone. It's very useful, although you often find subjects are unwilling to have their fingers stuck. Most common is serum collection, blood drawing, taking a blood sample. It's more invasive, a syringe or a needle, or sometimes an indwelling catheter. With an indwelling catheter, you can get multiple samples over a long period of time and really see the changes in hormones within the body. This allows collecting much larger volumes in humans. You can collect up to 20 to 40 mils of blood, which allows you to be able to assay multiple hormones from a single blood collection. In addition, because of the larger volume, it makes it possible to detect hormones that are in relatively low concentrations because you have such a large volume of sample. Hormones can be sampled from cerebrospinal fluid, so you can do a CSF tap. This little cartoon here shows a cross-section of a human. This is the spinal cord, and it's essentially bathed in cerebrospinal fluid. You do not need to know this term here. But essentially what it requires doing is sticking a long needle between 2 vertebrae in the spinal column and entering the needle into the cerebrospinal fluid that surrounds the spine, and then you withdraw sample, and you can get reasonable volumes of this. It has significant risks with it. If a person is not experienced and they actually stick the needle into the spinal cord, you can get spinal cord damage, paralysis in the worst case. Typically humans who have this often report a headache afterwards that as a consequence of removing the cerebrospinal fluid. The cerebrospinal fluid is replaced constantly in the body, but you can initially reduce the amount that's circulating until it builds back up. Sample volumes are such that it's possible to measure a number of hormones. In addition, it's often thought that the hormones that you measure in cerebrospinal fluid may reflect the actual levels of hormones that you would see in the brain, although that's not really clearly known for sure. The last way of sampling hormones are tissue biopsy. This is rarely used in humans, although sometimes it is. In some kinds of cancers where they want to see if the hormone levels within a cancer are there will be a tissue sample taken. Obviously this is the most invasive, and it's used to localize hormones within specific tissues, so you might be interested in the concentration of hormones in specific nuclei in the brain. or within specific glands. Historically, this has been used to look at, for example, the levels of hormones actually within the cells in the anterior pituitary, as opposed to the hormones that are secreted in the blood. There are a number of non-invasive methods. Probably the most common one is saliva. It has some real advantages. It's good for measuring steroids. They're easy to collect. Nobody objects to giving you their spit. They're thought to reflect the free or unbound levels in the blood. Remember I said that steroids that are attached to a binding protein can't go through cell walls, and the hormones in the saliva essentially have gone right through the cell walls and the salivary glands and into the saliva, and so the bound hormones are too big to get through there, and so what you get in salivary hormones is essentially a measure of free hormones. At least that's what's thought, but like most things it turns out it's more complicated than that, but generally it's a good proxy for free hormones circulating in the blood. It has another advantage in that the hormones in the saliva tend to vary in the same time frame as do the blood levels, so you can measure quick changes as quickly as you could measure them in the blood, which is a big advantage of saliva. The disadvantages are that it does not appear to accurately measure protein and peptide hormones. This is an area of some controversy right now. Some labs say that they can measure peptide hormones in the saliva, like oxytocin. Other labs say no, it's not possible to do. The hormones are in too low levels to be able to accurately be measured. It requires large amounts of saliva, from 5 to 10 mils, and that may not seem like much until you actually try and spit 10 mils of saliva, and it turns out that it's not that easy to do. The hormone concentrations, because it's often measuring the free hormone, are very small, which means the assays are technically difficult, and there's a lot of there's significant concerns about how accurate some of these assays are. This is an area of debate right now. The other thing about salivary hormones is that the hormones are affected by food, what somebody's whether they've just drunk coffee, for example, whether they've smoked a cigarette, whether they've drunk alcohol. There's a real problem with saliva in if you have any bleeding in the gums. If you've recently brushed your teeth, for example, there will be microbleeding there. The problem with blood contamination is that because the hormones now no longer have to go through the cell walls, you're actually measuring all of the hormones that are in the blood, and you get dramatic variation in the levels of hormones in the saliva if there's any blood contamination at all. Ideally, there are ways of checking for blood contamination in salivary samples, and ideally this is what should be done, but often is not. Because it's hard to get 10 mils of saliva, people often try to use things like gum to stimulate salivary release. This is controversial because there's evidence that these affect the amount of hormone measured in there without the hormone actually changing. It just inflates the values. One common technique has been used in animals and humans to use cotton balls which absorb the saliva, and then you can centrifuge the saliva out. And this seemed like a very good technique for doing it, except it turned out that cotton dramatically affected the steroid levels measured, particularly of testosterone. So generally now the requirement is to do unstimulated passive drool, which means basically you stick a straw in your mouth and you just drool in it until you get 10 mils of spit. Other non-invasive hormones samples are urine. This is wonderful because it's totally non-invasive and it's easy and quick to collect. You get very large volumes of urine, And the protein hormones that you see in the urine are the same as you see in the blood. The disadvantage is that the hormones concentrations vary with the time of collection. So, for example, first void, the first urination in the morning, essentially contains all the hormones since the individual went to sleep maybe 8 hours before. So within that urine is an accumulation of 8 hours worth of steroids, whereas the next time they urinate may be 2 hours later, and so actually there's been a much shorter period of time to collect the hormones in the urine. So it's very crucial to control exactly when the urine sample is taken. This reflects the fact that urine samples reflect the accumulation of hormones over time. They really cannot measure short-term changes like changes within minutes. They're not even very good at time frames within an hour. They're good for looking at the total secretion, the total excretion of hormones over a long period of time. And because they accumulate hormones over that period of time, even hormones that circulate in the blood in relatively low levels can often be detected in the urine because they're concentrated by the urine. The other difficulty for steroids is that the steroids in the urine are in conjugated forms and with very low levels of free hormones, which means to really accurately measure the level of hormones, the urine has to be treated in such a way that it's deconjugated so you can actually measure the specific hormones that are there. The other thing about urine is that the hormone levels that you see in the urine lag behind the blood levels by as much as 24 hours, which means that what you're seeing at first void in the morning really is reflective of some hormonal changes that may have occurred just an hour before, but also hormonal changes that happen 8 or 12 hours before. Fecal samples are actually relatively popular with field workers looking at animals in the wild where they can collect the feces from these animals. They're good for steroids and protein hormones if the proteins haven't been digested. They're easy to collect. I think people are not thrilled with the idea of collecting feces. I know an investigator who came back from South America with several hundred pounds of feces that they had collected. It can be fun going through customs. The nice thing about collecting feces, particularly for field studies, is they can be collected in the animal's absence. You can see them defecate, and then when they've left, you can go and collect it, which is nice for working with carnivores and other large carnivores. The disadvantages are that the hormones that are found in the feces are often degraded products. They've gone through the digestive system, they've gone through enzymes that are actually in the gut. The other thing is that once the feces have been deposited, you want to stop the enzymatic action, because if you don't stop the enzymatic action within the feces, they will continue to degrade the hormones. And so a real danger with fecal analysis of hormones is that if you don't control the sample really well, actually the amount of hormones will decline over time as they get broken down. The other thing about feces is that they're affected by a diet, that depending on what the animal is eating, the passage of material through the gut can be fast or slow, and that can dramatically change the amount of hormones there, because essentially what's happening is that the hormones are being collected by the feces as they pass through the gut, as the hormones come through the cell walls, and depending on how rapidly the feces pass through, it can very much affect the levels of hormones, which adds a lot of variation. They cannot be used to detect quick acute changes in hormones. They're like urine, they integrate hormone values over a long period of time. Other lesser sources of sampling hormones are sweat, exocrine gland secretions, and tears. These are used in very special cases where animals produce external pheromones. Although there is one study in humans that actually looked at changes in testosterone in tears in men looking at women under different situations. I'm not sure exactly what that study meant. The disadvantages of all these are extremely small volumes, can't detect hormones that are in very low values, and collection itself is very difficult, typically for exocrine gland secretion, sweat. Absorbent pads are used. The tears were actually collected in capillary tubes, but it turned out it was collected from people who were very good at crying. Factors that affect the hormones that are sampled. These are things that have to be taken into consideration when one collects their samples. For example, ovarian cycles. What can a single measure of estradiol, progesterone, or LH in a female tell us about hormones across the cycle? You can't just randomly go in and collect a sample from normally cycling females, because the hormones, as we know, are going to be at different levels at different times. So you actually have to have some way of estimating where in the cycle you're collecting the hormones. And if you want to know what the changes are across the cycle, you're going to have to take multiple samples at different times during the cycle. Similarly, what's the effect of being on a hormonal contraceptive in humans? This is a really important factor that surprisingly sometimes is not controlled for in studies. People don't actually state whether their women subjects were on oral contraceptives, on hormonal contraceptives or not. Circadian rhythms. Most hormones undergo a circadian rhythm. So you have to control for the time of day that the sample is collected. Typically the way this is done is by just making sure all of your samples are collected essentially at the same time of day. But things like long distance travel can alter the circadian rhythm, and the end result is that the individual will actually be seeing a peak in their hormones at a time when they were home would not be a peak in the hormones. So it's very important to be aware of the influence that circadian rhythms have on the secretion of hormones. The other thing is that many hormones are released pulsately. This is particularly important in gonadotropins like LH. They're released in 20-minute pulses, which means to really get a good picture of the sort of average level of LH, you need more than one sample so that you can average across the samples, because if you happen to take the blood sample right when there's a pulse, the level would be very high and then lower at another point. The last factor is seasonal variation. A wide range of species have seasonal fluctuations in gonadal activity. So in some species, gonadal activity ceases completely in the non-breeding season, and then they undergo essentially like a second puberty, and the system turns on again. So it's important to know whether the species that you're working with has seasonal fluctuations in hormones or not. How do we go about measuring hormones? Well, for most of the history of research in hormones and behavior, there was no precise way to measure hormone levels. Today, there are both qualitative and quantitative ways of measuring hormones, and we're going to talk about both of those. But there are many unsolved problems with hormone measurement. These compounds circulate in the body in relatively small amounts, and assaying them, it's quite remarkable that we can assay them as well as we can. But it is very difficult. There are three primary approaches to measuring hormones. The first one that we'll talk about is the bioassay, which is primarily a qualitative assay. It doesn't give you precise numbers of the amount of hormone, but just tells you whether the hormone is present or not. And bioassays allow you to look at a single hormone at a time. It can't look at multiple hormones within a single assay. The most common approach are immunoassays, which can produce both qualitative and quantitative results. They can give precise measures of the amount of hormone. They're very sensitive. They can measure hormones at very low levels. And again, they can do only one hormone at a time. The current method that's under development is liquid chromatography, mass spectroscopy. The tremendous advantage of this is that it allows you to measure multiple hormones in a single assay from a single sample. So you can, some assays measure up to 20 different hormones at the same time. Most of the work in measuring hormones has always been measuring one or two or maybe three hormones. Now we're entering an era when it may be possible to look at 10 or 15 different hormones at the same time, so you get a very different picture of what the hormones are like that are circulating. They're extremely sensitive. They're essentially measuring these at the molecular level. The end result is that they're more sensitive than the immunoassays. They're still under development, and I'm not going to cover details of this in the class. I think within the next 2 or 3 years this will become the most common way of assaying hormones, and is going to, I think, radically change the way that we look at hormones. But we won't go into that in detail in this class. We'll just talk about bioassays and immunoassays. So bioassay, it's actually the oldest form of hormone assay. The first published bioassay was done in 1860, so it's been around a long, long time. Bioassays were actually the way in which hormones were first identified. The importance of a bioassay is that it uses an observable biological response to a specific hormone. So it's a biologically based assay in that what you're looking at is a real-world biological response to the hormone. They're typically qualitative. They don't give precise values. At the end of this I'll describe a quantitative bioassay, but generally bioassays are qualitative, not quantitative. Here are some examples. Comb growth on a rooster, which is in which testosterone produces that. Here is a female. Here's a female with no comb, and here's the comb of the male. This is actually a bioassay for testosterone, and was the first demonstration. This was what was demonstrated in 1860. Was the first demonstration that the gonads of male roosters produced a compound that would cause comb growth. If a female rooster was injected with ground up male testes, she would grow a comb like the male does. So this was the first demonstration that there was something produced by the gonads that could produce this male-like characteristic. Another bioassay is uterine growth of the endometrium, which is a bioassay for estradiol. And in fact, this is used in humans a lot, in women, to look at whether treatments have peripheral effects on the uterus. We look at the thickness of the endometrium essentially as a bioassay for estrogen activity. Ovulation in the frog, which can be induced by LH or HCG, human chorionic gonadotropin, we'll talk about in a moment, has been a very common bioassay. And in fact, for many, many years, this was the pregnancy test. So if we have a woman's urine sample who wants to know whether she's pregnant or not, and we have a hypophysectomized frog, so we've removed the frog's pituitary gland, so the frog cannot produce any gonadotropins itself. So this is a female frog, but she has ovaries, but she has no eggs in the ovary. If we take that urine sample, if the woman is pregnant, her urine would contain human chorionic gonadotropin, which is produced by the developing placenta in the fetus, and HCG, which is very much like LH. It's a very similar construction to LH. Can stimulate ovulation in frogs. So with our hypophysectomized frog who has no gonadotropins of her own, if we inject her with the urine sample, then if it contains HCG, she should ovulate. If not, she won't. So if we see that she has no eggs after several days, We know that the woman is not pregnant because no eggs were produced. But if eggs are produced, then we know that the urine had HCG in it, and the woman is pregnant. So this was the typical pregnancy test for many, many years. It took at least 3 to 5 days before you could get an answer. It had to be done relatively late in pregnancy, not late, but certainly 6 to 8 weeks after pregnancy had actually occurred was the earliest that it could be detected with this. It's not particularly a sensitive assay. And the problem with it, it's also subject to false negatives and false positives, so it's hard to tell. They often had to be run twice to be accurate. This is an example of a qualitative bioassay that uses a biological response, in this case HCG inducing ovulation in the frog, to determine something else, whether a woman is pregnant or not. So, to summarize, bioassays are typically qualitative, ovulation or no ovulation. They always reflect biological activity, which is important because this is actually the only assay that truly shows biological activity of the hormone measured. Since it relies on a biological response, you know that the hormone is biologically active, unlike some of the other assays. The biological endpoint must be dependent only on the hormone to be assayed. If it's dependent on multiple hormones, you won't be able to get the response. And as we'll talk about later, new techniques have actually produced quantitative bioassays that still rely on a biological response. Okay, how about quantitative methods of measuring hormones? And in this category are immunoassays. Immunoassays take advantage of the immune system's ability to create antibodies. So one of the large systems in our body is our immune system, which protects us from a variety of diseases. And one of its primary ways of doing this, among others, is the creation of antibodies. The immune system recognizes any proteins in the body that are different from self. So it can identify foreign proteins. And so, for example, when you get a virus, it has proteins that your body doesn't have. Your immune system recognizes that, and it triggers an immune response, and the body then proceeds to create antibodies that bind to and inactivate those foreign proteins. So that's how the polio vaccine or measles, mumps vaccine, any of those work, that essentially they introduce into your body foreign proteins that cause you to form antibodies. And long after that protein's gone from the body, you continue to have those antibodies, and it's a way of fighting disease. So how does this immune response get initiated? Well, we need to talk about a number of different aspects. So we have an antigen, and that the antigen is what binds to the antibody. And the antigen is typically the protein that we're talking about. It's what stimulates the immune response. It's what the antibody is formed to. It triggers the immune response. But an antigen can have multiple sites on that antigen that can stimulate the formation of an antibody. And those sites that can actually trigger the formation of an antibody are called epitopes. So in this antigen here, this protein, I showed that it has 3 different epitopes, and that it ends up triggering 3 different antibodies, one to each of those epitopes. The antibody for a given epitope is specific for that epitope, so that's where it will bind on the protein, and that's what triggers the immune response. So here you can see we have 3 different epitopes. Here are the 3 different epitopes here, and we get 3 different antibodies, each with a different kind of binding site that is tailored to that epitope. So, how are specific antibodies produced? Well, you inject a foreign protein or peptide into a species. For example, sheep and rabbits are often used to produce antibodies. That's different from the species to be studied. So if I'm making an antibody to LH, that I want to measure LH in humans, then I can inject LH into the sheep. The sheep produce an antibody to that. The immune system recognizes that the injected protein is foreign and creates antibodies to inactivate it. Then those antibodies remain in the blood and they can be harvested, so you can actually separate them from the blood and collect them. And then those antibodies can be used in immunoassays. That works fine for peptide and protein hormones. You simply inject the hormone into the body and collect the antibodies. But there's a problem with steroids. Protein and peptide hormones easily produce antibodies as these compounds trigger an immune response on their own. But steroids aren't proteins. As you know, they're much smaller than proteins, and they certainly aren't proteins. They're not peptides. Steroids are the same in all species, so there is no foreign steroid. So if I take testosterone from a rat and testosterone from a monkey, your immune system is going to see both of those as exactly the same compound. In fact, it's not going to do anything because it's not a protein. It's not going to trigger an immune response. So how do we get an antibody to steroids? So what you do is you can create an antibody to steroids by linking a steroid to a protein. Typically something like bovine serum albumin is used, but you take a neutral protein that's not a hormone itself, and you link the steroid to it. So now you have a steroid protein complex, and the steroid molecule that's attached to that protein becomes an epitope on the protein. Remember, an epitope is the site that the antibody will form to recognize, and it will bind to that epitope. So by combining a steroid molecule with a protein molecule, you create an epitope on that protein now that is unique to that steroid, and that protein steroid molecule becomes the antigen, triggers an immune response, and you end up getting a antibody that's created to this foreign protein that's specific for the steroid used. It'll probably produce antibodies to other epitopes on that protein, but what we're interested in is that it will produce an antibody that's specific to that steroid, and then we can harvest that, and we now have an antibody that recognizes the steroid, even though the steroid is no longer bound to the protein. It will actually recognize the steroid free floating in the body. Just a reminder, the epitopes are the parts of the antigen where there are differences between self and foreign proteins, and that once the antibody has formed to the steroid epitope on the protein steroid complex, that antibody will always recognize the steroid even if it's no longer bound to the protein. So, to do an immunoassay, what we have to do is we have to determine whether the antibody has bound any antigen, the hormone in this case, and in what amounts, how much of the hormone is present. That's our goal. So one way to do this is to tag either the antibody or the antigen with something that can be seen. I mean, these are small compounds, and so we can't visualize them, but we can give them a tag that allows us to see where either the antibody or the antigen is. So the tag could be radioactive, or the tag can be an enzyme that produces a visual change, typically color upon binding. So those are two mechanisms to make them visible. One, radioactivity. We've got to have some way of counting radioactivity. We can't see it. In the enzymatic assays, we have to have a specific way of looking at color change that reflects the amount of the hormone. So we get different patterns of binding depending on whether the antibody or that's supposed to be antigen is tagged, and we'll talk about those in detail. So let's start first with a competitive assay. This is an assay in which the antigen, in this case the hormone, in this case a steroid, is labeled, and this is a radioimmunoassay, or often referred to as an RIA. This is probably the most common form of immunoassay that was done, although in recent years enzymatic assays are becoming much more popular because they don't produce any radioactive waste. And I would anticipate it won't be long before radioimmunoassays are only used in very specialized conditions. But most of what we know about hormone measurement originally came from radioimmunoassays. So here I'm going to take you step by step through a radioimmunoassay and how it is produced. And this is somewhat complicated, but not that complicated. So this is my sample, and these little purple dots here, oops, I realize I'm not showing. So these little purple dots represent the hormone that I want to measure, and here you can see it's got the little epitope on it that's where the antibody is going to bind. And this sample has more or less cold hormone. That just means the hormone that's circulating in the sample. This could be a sample that has no cold hormone in it because the animal wasn't producing any hormone, but this could have a lot in it. So this is the hormone sample to be assayed. This, in the laboratory, I take some of the same hormone and I attach a tag to it, in this case a radioactive tag. So these molecules actually have a little radioactive tag that these molecules don't have. So this is the hot hormone, and I know exactly how much of that I put into the assay. This is the antibody. And notice the antibody has a site that looks like the site, the epitope on both of these hormones, which means this antibody is capable of binding either or both of these hormones. It doesn't make any difference whether it's got the label or not, the antibody is going to bind it. And I put in the antibody, I know how much antibody I put in there, so I know how much binding can actually happen. So I combine this all together in a medium, and I let it incubate for some period of time. Typically now it's in a matter of hours. And then what I want to look at is, go back, that didn't bind because there wasn't enough antibody to bind everything. So think of this like I have this big soup that's got hot and cold hormone, and what the antibodies do is they just reach into that soup and they take out a sample of it. It's essentially a random sample of it. It would be like if I had a bag full of yellow and black marbles in different proportions, and I shook them all up, and then you reached your hand in and you took 8 marbles out. That would be a random sample of that, and the proportion of yellow and black marbles that you got from that random sample should be very similar to what the overall proportion of black and yellow marbles are in that bag. So it's the same thing here. The antibody binds both the hot and the cold hormone, and it takes a sample of it out, and the proportion of hot to cold should be the same as it is in the overall sample. I'm just looking at a subsample of it. And so here's what I end up with, my estimate here, and now I can count how much radioactivity there is in that sample to tell me how much hot or cold there is. Because the basic principle is, if there's a lot of cold hormone, in this sample here, then very little hot will be bound because most of the sites will be filled with cold hormone. If there's very little cold hormone, then a lot of these sites are going to be filled with hot hormone because there was proportionally less cold hormone. So when I count this, I take this now and I count it. Let's turn to an immunometric assay. This is a double antibody assay. And in this case, instead of labeling the antigen, or the hormone, we're going to label the antibody. And this works because we're going to actually have 2 antibodies in this. So I have here, this is the glass wall of my test tube, and I have an antibody that's actually been bonded to that tube wall. It's stuck there. It can't go. It doesn't go into solution. It's stuck to the wall of the tube. These are called coated tubes. They're coated essentially with antibody. And notice that it has a binding site here that's specific to one of the epitopes on this hormone, the little diamond-shaped epitope. Here's my hormone. It has two epitopes, and I've created two antibodies, one to this epitope and one to this epitope. This second antibody is labeled, and here I'm just this is going to be a little color label here. So this antibody actually is colored, and when I incubate these together, what I get is the formation of this sandwich. So this hormone acts as the glue to bind these two antibodies together, and so now I get this color signal on the side of the test tube that's there because the hormone has bound those two antibodies. Now if there is no hormone in the sample, these won't bind, and when I separate this, in this case these stay behind the tube. Anything that's not bound to this second antibody is going to be poured off. It's going to disappear, and it won't leave anything behind. So I use a special device that can actually count it. It's not truly a counter. What it's doing is it's looking at color change, and it can tell me how much color is left behind of the color that I'm looking for is left behind in this sample. What's important about this kind of immunometric assay with a double antibody assay is that the intensity of the signal, in this case it's not radioactivity, it's color. The intensity of the signal is directly proportional to the amount of hormone. If I had no hormone, I wouldn't have any signal because none of the second antibody would actually get bound and stuck to the wall. If I have a lot of hormone, then I'm going to get a lot of signal because all of the antibodies that are attached to the wall are going to be bound to another antibody that has the color signal. So the signal is directly proportional to the amount of the unknown hormone. And in fact, if we compare these two kinds of systems, if we look at the relationship between cold hormone and the amount of signal determined by what is labeled antibody or antigen, if we label the antigen in the competitive RIA that I gave you, as the hormone concentration in my sample goes up, the percent bound radioactivity goes down, or in other words the signal, which in this case is radioactivity, declines as a function of the hormone, increased hormones. In the immunometric assay, sandwich assay, where the antibody is labeled, essentially the color intensity increases with hormone concentration, so it gives me a direct relationship. So, issues that we need to remember when we think about hormone assays is that hormones that have been degraded, in other words they're biologically inactive, may still bind antibodies. If that epitope is still available for the antibody, it can still bind the antibody even though the hormone no longer has biological activity. So the binding would suggest biological activity where in fact there really is no biological activity. So here's our antigen with its three epitopes and the three binding sites. Let's say we degrade it. So now I've essentially lost a large portion of this compound, but I still have two epitopes here. The end result is that I could get continued binding of this, but it wouldn't have any biological activity. It's in that color spot are a whole stockpile of colored antibodies. So they're hidden under the plastic shell, and they're specific for one of the epitopes on HCG. Where we saw the thing that said the test line is actually a whole line of antibodies with nothing attached to it. And you can't see them, they're invisible because until the colored antibody binds to them, you're not going to see them. Just as you can't see the control line either. It's the control line is a row of antibodies, but they actually already have some HCG bound to them. and we'll see why that is later. So this is the way the system looks before we introduce a urine sample into it. We have a supply of the second colored antibody. We have the first antibody in the test line and the first antibody in the control line that already has some HCG bound to it. So what happens when we put a urine sample? So here's the urine sample. It goes up along the wick. This whole thing is actually sitting on a kind of wick that wicks the urine through first the pool of colored antibodies. It picks them up and it carries them along, and now it reaches that first line. But because in our sample here we have no HCG, there's nothing to bind this blue antibody to the green antibody. So our sample just keeps going on through until it hits the second line, and the sample continues on, and what's left behind are all of these antibodies that now bound the HCG that was already there. Right? So what I would see in the window is I would just see the control line would be colored, in this case blue, and I wouldn't see the test line at all because nothing bound there. And so the woman is not pregnant. Since there's no HCG in the sample, the colored antibody only binds to the reference or control line, so one line appears in the window. So now what about a sample where the urine sample contains some HCG? The woman is pregnant. Now when it goes through that pool of antibodies, you can see some of these actually bind to the epitope of the HCG, so it picks up some of those antibodies. As it travels through the test line, it actually binds to that test line because it binds to the other epitope. This epitope binds to the colored antibodies, and the end result, right? In this case, it's binding to the HCG that was in her urine, binds these together, and in this case, it's binding to the HCG that was already attached to this. So why do you think they have the control line? Well, this is a way of warding off possible false negatives because the system didn't work. You always have in the test line, in the control line, you always have an example that yes, the pregnancy test is actually working, because you should always get at least one line. If you don't get any lines showing, then it means the test is no longer functional. And because because you get the control line, you know that the colored antibody is capable of binding to HCG. So if there was HCG in the urine, it should have showed that up on the test line. Last thing is, how do we make a quantitative bioassay for LH? So there's really a practical reason for this. In some species, the LH immunoassay is problematic, and that's because the epitope that produces an immune response or results in the creation of antibodies doesn't actually predict the hormone's biological activity. So we have an antibody to LH that definitely identifies LH, but it can't tell me whether the LH is biologically active or inactive. So the way to approach this is to make a quantitative bioassay. And what we're going to do is we're going to use mouse Leydig cells for a quantitative LH bioassay. Why Leydig cells? Well, because Leydig cells produce testosterone when they're stimulated by LH. So if I take Leydig cells from testes of mice and I put them in a tissue culture like this, disperse them and I put them in a tissue culture, if I put my sample that I think might have some LH in it into this culture medium and I incubate it, and this takes hours to do, these little cells, I'm sorry, I keep forgetting that I'm not showing, put it in this culture medium and I incubate it. These little Leydig cells, if there is LH in this sample, should start releasing testosterone into the culture medium. So I let this incubate for a number of hours, and then I remove the culture medium, and I then can take that culture medium and I can assay it in an immunoassay, for example, which is very precise and quantitative, but I'm now assaying for testosterone. And I can measure the T release from the Leydig cells in the medium with an immunoassay, and that gives me a precise number of how much testosterone is produced. And the only way that testosterone would be produced is if there is LH in this sample. So I'm using a true biological response, which shows that the LH in that sample is biologically active. And I'm using the testosterone assay to be able to quantify it. So this is actually one of the very best ways of measuring LH, because I know that what I have is a biologically active form of the hormone, but I'm not actually measuring the hormone directly. I'm measuring it indirectly through its ability to cause Leydig cells to produce testosterone, which then I do measure.